Okay, welcome everyone for being here. Today I want to talk about a project that is like a soap opera-like that got me interested in the last couple of years, that is exploring extra-pair paternity in this bird, the Louisiana water thrush. So many species of songbirds nest in pairs, right? And these social living arrangements, either because they share a territory or because they care for the nest in that territory, without inferring any sexual patterns or interactions, is called social monogamy. And until the 1990s, we thought that some birds that nest in pairs were always like a faithful father with the mother working together to rear their offspring. But we learned since the 1990s that that's most of the times not of the case for some families. Actually, it's what we consider socially monogamous species tend to play a lot of away games. And that's what gets the gossip out of today. So that's why we coined the term reproductive or genetic monogamy, where that's the case where we have a father and a mother always mating together and caring of their offspring. So understanding the genetic mating system in any species is important to understand the evolution of its populations, but also what are the drivers of natural selection and sexual selection. As I mentioned, in recent years, it has become common known that genetic polyandry, that is when the female mates with more than one male, is quite common in birds. And also genetic polygyny, that is when the male mates with more than one female, it's also present, but it's less frequent than polyandry. So, for males, genetic polygamy or polygyny, it makes sense evolutionarily speaking. Like they can mate with as many females as they want, and they can produce as much offspring as they can, but then they don't need to incur in the cost of taking care of that offspring, right? However, the adaptive benefits in females are widely misunderstood, right? Why? Because for them to go to mate with another pair that is out of their social pair can increase the risk of predation, can increase the risk of disease, can even make that the loss of the paternity for her nest and care. So why are females getting into this mess to start with? So a lot of the hypotheses according to this are hypotheses either adaptive, that it has an adaptive component, or either ecological, like the chances based on life history traits or territory or get there, and she just uses for taking care of those or incurring in these polyandry events. In this study, we wanted to study the Louisiana water thrush, that is a small stream obligate bird. What is really cool about this bird is that they nest along linear territories, and that gave us a specific opportunity to understand who's the neighbor, how big their territories are, in a quite easy form. Now, we wanted to understand what is the genetic system of the Louisiana water thrush if extra-pair copulation or extra-pair paternity, that is polygyny, polygamy, are present, and if it's present, what's the frequency of these events? But also, maybe what are the drivers for the species based on the kind of data that we could collect? So we are going to test some ecological, but also evolutionary drivers. So throughout 4 breeding seasons from 2009 to 2012, my colleagues collected and monitored water thrush birds along 4 streams in the Ohio River drainage. These streams were purposefully selected based on water quality. So the 2 in the south, I guess I have a pointer here. So these 2 that you can see towards the south tend to be higher quality streams versus the ones that here are more towards the north tend to be lower quality streams where anthropogenic runoff and agricultural activity are the main things affecting water quality. So the Louisiana water thrush in these study areas were target sampled by mist nets and sometimes playing calls to try to aid with capture. Adult sex was determined for all adult birds, and we were able to systematically search for all nests that we could find. So when we see a pair, we try to find everywhere where the nests are, right? And we were able to also identify who were the social pairs. So who are the pairs that I'm seeing in the field are always together taking care of a nest, right? So we took DNA samples for both adults and nestlings, and we extracted the DNA and did 3 rat libraries. This sequence data was then utilized to do a genetic diversity and connectivity analysis that can help us to make sure that we fill the assumption of panmixia, so we can do some relatedness analysis. And for that, we can then test for likely drivers for the EPC. Okay, so here are the results. Here's the gossip part. I hope I haven't spoiled it too much. We saw in the field 317 individuals, right? And 50 nests, successful nests. Out of these, we collected samples for 288. What is really important is that in the field, we didn't see any attempt of infidelity. We didn't see any attempt of EPC happening in the field. We did see, although some cases of divorce where the pair change its mate in the next breeding season. And that brings me to a topic that I'm defining polygyny and polyandry very strictly here. I'm only defining them when they happen within a breeding season. If a pair changes its mate between breeding seasons, I'm calling those divorce. So it's very different from polyandry and polygyny, the terms I'm studying here. So we use our sequence data, we use our rat-seq data and map to a reference genome of a closely related species as the common yellow throat, and we got thousands of loci, right? From those loci, we only use those that are in autosomes based on the reference genome, and only SNP per locus, and we calculated 3 kinship coefficients that are listed in this slide. The good news is that those kinship coefficients were very similar between them with high Pearson correlation coefficients, very significant. And the cool thing that we found is that, yes, all offspring within one nest has the same mom. So all of them come from the same mom. And one other cool thing that we found is that adults almost return to the same stream between breeding seasons. So there's a little bit of a phylopatric behavior there. Only one exception was seen, and that adult divorced its mate between breeding seasons. Then what we did is that we took our 48 nests that we collected genetic data for. Out of those 48, we can only assess 47, and we assess are there half-siblings inside that nest, right? So what you can see is the orange represents half-siblings relationships, and we were able to conclude that 12 nests out of the 47 that we had extra pair offspring inside that nest. So it's important for you to realize that an extra pair of offspring inside a nest that share its mom is that the mom is the one that is playing some away games here, right? So, and this was observed for all streams. Now, we got that data and we try to make what I call the familial groups, that is, try to make these networks, or if this bird is related somehow to this other bird, how does that look like for all the streams? 10 of those, out of those 19 familial groups look something like this, like a monogamous relationship where always the male and the female, no extra pair of offspring are observed. We do observe some cases of double brooding, that they don't lay just one nest, but they have actually multiple nests within a breeding season, but all the nestlings belong to the same parents. Or sometimes we see that faithfully coming back between breeding seasons and always the same pair mates. That is the example that I have here. However, the other 9 out of the 19 familial groups that we see are quite complex, and I'm just presenting you an example here. So these are birds from just one of the streams, the powder mill run, and what this very complex figure represents is that we observe cases of polyandry, where one female mates with one other male in the same breeding season. Cases of polygyny, when the male mates with more than one female also in the same breeding season, and actually this male doesn't mate only with one female, but 2 females inside the same breeding season. And we see those cases of divorce between breeding seasons. So what is really cool is that this is the T. Like, they do commit polyandry and polygyny in all streams. Now, how frequent is that? Polyandry was observed 12 times across all streams, and those represent these 10 familial groups. And as I mentioned, most of the time it's just one extra pair male, right? On the other hand, polygyny was less frequent. If you count the double extra pair mate, it was observed 6 times in the whole stream, so polyandry happens kind of twice as often as polygyny. And you can observe that we were like, when polygyny happens, there's always one nest that is pretty small, so only one nestling here. So we kind of analyzed if there was any indication that the males were incurring into polygyny because they detected that a previous attempt to reproduce was penurious. But we don't see any evidence for that based on when we collected the nest. Sometimes the nests were collected, the big nest was collected after the smaller nest, but sometimes the big nest was collected before the smaller nest. So we don't find any evidence for that happening. So what are the potential drivers for the extra pair copulation that we're seeing? So there's some hypotheses. One of them that you may know is the good genes hypothesis, where females are trying to maximize the survivorship and reproductive success of the offspring. However, we don't have that kind of data in this study, so unfortunately we cannot assess that hypothesis. But these other three we can assess at some extent. So first is the genetic diversity hypothesis, where females are seeking to maximize the diversity among their offspring. So we kind of first approach this as, let's count the number of alleles in nests that have extra pair offspring and nests that don't have extra pair offspring. Do they tend to change? The answer is no. So far, nothing there. What about within a nest? Do the offspring reared by the social mate have lower heterozygosity than the offspring reared by the extra pair? We calculated allele richness and observed heterozygosity. The answer is no for allelic richness. There's no differences. For observed heterozygosity, we observed differences, but not in the direction we were expecting. Actually, the social pair diversity was higher than the extra pair diversity. So again, we don't have any evidence for the genetic diversity hypothesis. Similar to this hypothesis is the genetic compatibility hypothesis, where females are trying to find very far distant, non-related males to mate to maximize their heterozygosity. And again, we wanted to compare between nests that have EPO, extra pair offspring, and nests that don't have EPO to see if the observed or even expected heterozygosities might differ, and the answer is no. And I was like, okay, what about if I compare the kinship coefficients in those pairs that have polyandry or have polygyny, and with those kinship coefficients between pairs that don't have these? And the answer is no. Yeah, so I'm spilling the tea, but not the full tea. So there's the fertility insurance hypothesis, where they think like she's also maybe in an attempt that her social mate has low fertility, she's trying to find mates with higher fertility. Based on broad sizes, we don't see any pattern, but we still need to do some statistical analysis there. Now, jumping to ecological drivers that we're testing in our study, the first one that jumped to our eyes was habitat quality. That's why we collected in such habitats. The reason behind this is that birds that tend to live in low-quality habitats tend to have longer territories. And we feel like the territorial behavior of males and females can be not that efficient when you have a long territory to care. So they have more chances to incur into EPC. The problem is that we didn't see any effect there either. So up to the moment, we don't know for sure why females are going into this mess. There's some other drivers of EPC that we can test that are explicitly testing territory length, explicitly testing if the age. Are females going for younger males? We'll see. Sex ratios, if there's skewed sex ratios within an stream, is that what explains the higher ratios of EPC or density? And why not neighbors versus bachelors? Who are the females cheating with? Are they cheating with their neighbors, or are they cheating with bachelors that don't have a set territory? And another one that I like to test, that is what I call sweet revenge, is polygyny happening because polyandry happened, or polyandry happening because polygyny happened. So far, my conclusions are that more often females than males find opportunities to commit EPC, but we know that both exist in the Louisiana water thrush. And with that, I want to thank my co-authors and universities and funding sources for this project. I hope you liked it. Yes?
In the cases where there could be males mating with multiple females, do you ever see anything with like male competition or sperm competition, how that might impact offspring?
Yeah, so we didn't assess for male competition in the field just because in the field we didn't observe this extra pair. Like we never guessed that. So it's true that sometimes we saw the other bird in the field coming to that territory, but we never saw them in action, and we never saw them that close to the male or that close to the female to estimate aggression. And in terms of a sperm competition, we don't have that kind of data, but that's a very cool hypothesis, yeah.
Hi, everyone. Thanks for coming to this session. My name is Rilquer Mascarenhas. I'm a PhD candidate at the University of New York, and I'm actually really excited to share some initial results that I have for one of my chapters in my dissertation, and I'm going to try to be by the microphone but also see my slides. It's a PDF, so it doesn't show up here. And in this chapter, we try to think about life history strategies and how to incorporate them in genetic models of demographic history. So we all know that genetic data allow us to estimate population. I think this is going to be better. I need to see the slides to see what's going on. Thank you.
Yes.
Okay, we all know that genetic data allow us to estimate the population demographic history because genetic diversity and the patterns that we see are correlated to affect population size. And a big way that we do that is by using simulations. So we draw from population genetics theory to create what we think is expected of genetic diversity under different demographic scenarios, and then we compare that to what we have in observed data and make inferences about species. And the big thing that in comparative phylogeography we do is that we make inferences across several species that are distributed in the same area, and we try to think about how they have responded to the same environmental changes that have happened. And the big thing we see is that species differ a lot in nature about they have responded to environmental changes that happened in the past. The main reason that people bring up about that is that ecological difference are commonly used to explain this idiosyncratic response, and that is counterintuitive. So if you think about species that have different habitat associations, you'll think that they, even if they are in the same environment, they are going to be occupying different parts of the environment, so they might have different effect population size, and the pattern of genetic diversity is going to be different when that environment changes. Same thing can happen when you have species that have different intrinsic dispersal capacities. You have species that are occupying the same area, but they disperse differently because of the morphology or because of any other ecological feature. The connectivity of populations are going to be different, and when environmental change happens, you're going to see different patterns, right? So the interesting thing is that we're trying to bring those things into the simulations that we do, so we can kind of tease apart if species are different in the patterns because they had different histories or because they have the same history but respond to that differently because of their ecology. A big part of their ecology that is a little bit harder to incorporate in simulations is what we call life history strategies, which is kind of matches what we've seen in the previous talk. And life history traits and any traits are related to the reproduction of the species, so things like the fecundity rate, the mortality rate, the age of the individuals that are reproducing, how many mates you have every season, if you're forming more monogamic pairs or polygamic pairs and stuff like that. From population ecology, we kind of know that these things will influence the number of individuals that are reproducing in a population, the number of offspring that is generated. So the overall population dynamic is going to be different depending on your life history strategy. And from population genetics, there is a lot of literature showing that they will influence affect population size, especially in species that have overlapping generations, right? And if they do influence affect population size, they will probably influence the patterns of genetic diversity that we have at the end. Three key life history traits have been brought up a lot in the literature to talk about the influence of genetic diversity. The adult longevity, how long you live, the age that you start to reproduce, and the mating system, if you're forming more monogamic pairs or polygamic pairs, right, and any range in between. And the literature says that the way that these life history traits might influence affect population size and genetic diversity is basically by influence the variance in the reproductive success across individuals, which basically means you have a gradient of this variance where if you're very low variance, it means that everyone has the same opportunity to reproduce, so all individuals are kind of reproducing every time. And if you have a higher variance, there's something happening that some individuals only reproduce, and there are many individuals in the population that are not reproducing. And when it comes to life history traits, you expect that lower values of those three traits that I was talking about before would be related to low variance. So species that are short-lived, that start reproducing right away, and that tend to form monogamic pairs will tend to have low variance. And the opposite would be expected in species that have higher values for those parameters. They are long-lived, they start to reproduce a little later, and they tend to form polygamic pairs. So you have many individuals that this would be the mating success, some individuals that happen here, and a lot of individuals that don't get to reproduce, so you increase the variance in the success that you see in the population. And those are expected to have influence on genetic diversity because you expect larger population size, shorter generation time, and a higher genetic diversity on the low variance end, and the opposite on the high variance. So these kind of things are we're trying to think about when incorporating simulation to start inferring demographic history with those things in mind. And the first question that we're thinking about is how those life history traits influence generation time, the variance in the success of reproduction across males in case of polygyny, and genetic number statistics, and can life history differences be inferred from genetic summary statistics? Do we have the signal of the ecological difference across species when we look just at genetic data, and also across different demographic scenarios? So just think about that. We are implementing a bunch of genetic simulations with varying these life history parameters, and we're exploring the results of this variation on generation time, variation of success to reproduce, and the genetic summary statistics that we get at the end, and to quantify the signal that we have from genetic data to infer ecology. We're doing a quick random forest model to quantify if we can estimate the parameters of how much variance you have in that success, and if you can choose and if you can correctly identify the demographic model that generated that genetic data, right? That's the methods. Because these things are kind of complex to simulate, we are jumping to individual-based models so we can control the behavior of the individuals. You can tell them when they will reproduce, when they will not, why they will not reproduce, if they're going to form monogamic pairs or polygamic pairs. And basically, we are doing a model that has two stages, reproduction and death. And in the reproduction, you have them mating and generating offspring, and in the death, you have offspring mortality and the adult mortality and the carrying capacity acting. And basically, our three parameters that are interested are the age of first reproduction, and the number of mates that you have that go in that first stage, and then the longevity, how long you live that go in the death stage. And what we're doing is that we just we are varying the values of those parameters and making all those combinations on SLAM, which is a 4 genetic simulator, and we're doing 100 replicates for each of those combination of simulations so we can explore the behavior. So SLAM is giving to us a couple of ecological dynamics of this population, the variance in the success rate, the generation time, the number of individuals that are actually reproducing, and a really cool thing is that SLAM is keeping track of the genealogy of individuals. So we get to simulate the whole ecology there, and then we get those information and jump to a coalescent model that adds mutations to it. We coalesce the genealogy, we add mutations with mutation rate and population size, but they are informed by the ecology of the species that is varying depending on the combination of parameters I'm doing. And then we do genetic summary statistics, and we did 4 of them: height, the GMC, the number of segregating sites, and the allele frequency spectrum. Next. Random forest models is what we're doing to quantify the predictive power from genetic data. They're really cool to work with a lot of data and with a lot of predictors. When your goal is to predict and not to understand and explain, they're really nice for that. And so we're doing 2 kinds of model. The first one is we're focusing on seeing if we can estimate the variance in the success rate from the genetic data. So we're doing a regression model in the forest, 1,000 replicas to quantify this model accuracy. At each replicate, we just set aside a part of the data set of all the simulations that we did. And we show the model a set, teach how to do it, and then we ask it to evaluate on the other set and see if it's doing well. And then we see how well the model is doing. And in the second model, we did, next one, we did another set of simulations where we focus and simulate a monogamic species, a polygamic species. So the monogamic should have low variance, the polygamic should have high variance in the success mating. And for each of these species, we did 3 scenarios: a mictic population with no vicariance, and a vicariance with low migration between them, and a vicariance with high migration between them. So here are we asking, the model can tell apart species that have different ecologies and the histories that they would have, right? And the results are brief and preliminary, but basically our first result is exploring how the life history parameters are influencing a couple key ecological parameters of the population, and the genetic diversity is the first one that we're thinking about when looking at genetics. And if we go one by one, we see that when we look at the variance, the main one that influences the number of mates. When we look at generation time that comes from SLAM, all of them, it's longevity and age of first reproduction. Number of mates, a little bit, not as much. But most of the parameters that are affecting age are the ones that affect generation time. And combined, number of mates and age of first reproduction seem to decrease a little bit, affect population size and genetic diversity, and longevity does not have that much of an effect. So those are interesting things to think about when you're looking at literature about overlap generation. Think what parameters are actually controlling genetic diversity at the end, and where's the signal of the ecology affecting genetic diversity. And for the second question, next slide, this is like a histogram of all the R-squares of all the 1,000 replicates of rainforest models that we did. And they mostly fall around 0.4, which can be considered low, but for rainforest is kind of really good, considering how much biological noise you have and how you don't have that many predictors in here when you think about the summary statistics. So there are other models that are like okay in this range. So what we're thinking is that there is some signal there. It's just to think about where the signal of the ecology is going to genetics and what summer statistics can capture that more. And this is where we're trying to see different demographic scenarios. This is a confusion matrix that should have 1, 2, 3, 4, 5, 6. So we have high variance species, the polygamic species, and the low variance species. And here you have the same thing, the high variance and the low variance species. So it's like every time the model was a low variance model, the species was a low variance species, it was a monogamic species. My rainforest model, how many times could it tell it was a high variance species, right? So you want to see the diagonal be more red because it shows that the model is getting it right. And what we see is that the model can tell apart monogamic versus polygamic, but at this point it's not being able to tell apart migration. And I believe it's just related to some tweaks in the simulation. I think it has to do with how long we're simulating things and letting the genealogy show it before we coalesce things. But so those are preliminary, and we have to think about that more. Next. So yes, I skipped the one part. So basically, we have a couple take-home messages from this preliminary results. You have reproductive age and mating system is the main thing that influence the variance in reproductive success, mostly mating system, but we see that the age that you start reproducing also decreases the type population size, so many individuals do not get to reproduce because they didn't reach the age yet. Generation time is heavily influenced by longevity and the age, but mostly the latter, the age that you start to reproduce, is the one that leads to lower genetic diversity because it decreases, again, the amount of reproducing individuals. Next. The genetic summer statistics do vary with life history parameters, but the accuracy of the parameter estimation is moderate. And the next one. Go back.
Sorry.
It's okay. We can tell apart ecologies, but we cannot tell apart history yet. That's basically what we're getting from this. Big thing that I want to try to do next is to get data from a polygamic species, or the one that tends to polygamy, the one that forms territories. There's very few mannequins, they do get this thing of they help each other, the males, but not all the males in the group that is helping each other get to reproduce, just the alpha male to different degrees in different species. So they do have this variance higher. I want to see if from the genetic data, just get all the sum stats I can and train my model with simulation and see if I can see, yes, there is higher variance in that species. And the next step is to make everything spatial, if possible, because we know that everything is influencing ecology. So it's an interesting question to tease apart in what species the life history is going to be an important factor to predict the story versus in what species morphology and dispersal capacity is what is important versus in other species ecology, the intrinsic phenotype does not really matter, but it's mostly the environment and the climate and the topography that is influenced. So teasing apart these three sources of genetic variation would be very interesting. That's it. I would like to thank
All right, tēnā koutou katoa. Nice to meet you, everyone. I'm Natalie Forsdick. I'm a researcher from Manaaki Whenua Land Care Research based in Aotearoa, New Zealand. I really wanted to give the auto captions a bit of a run through there. We're dropping in some Maori terminology along the way. I'm really excited to be presenting today on a fairly simple story about some conservation genomics work for a little seabird that occurs only in New Zealand, the whenua diving petrel. This seabird is a critically endangered seabird. They're very small. They were once widespread across the southern South Island of New Zealand and some of the small offshore islands down the bottom. New Zealand is a really small series of islands, and currently the whenua diving petrel is only found on one of those very tiny offshore islands. And not only that, but they only occur in this one single strip of dune along this beachfront on that island. So this is a smaller area than a football field, for example. They're very, very restricted in their breeding range. And along with that, there's only 200 adults remaining at this point. So they've got all the small population challenges that they're facing. And they also hybridize with the common diving petrel, which, as the name suggests, is a much more common, widespread species across the Southern Ocean. So they've really got their work cut out for them at this point. Just to talk a little bit more about that hybridization, we always see male kuaka pairing with female common diving petrels, and of the eggs that are produced, about 95% fail to hatch. So that's we're seeing some definitely not hybrid vigor going on there. In terms of those offspring that do manage to hatch, we see some morphological characteristics that make them probably definitely less fit than either parental type. So whenua diving petrels have these amazing traits that allow them to do some awesome stuff down in the very fierce, rough, cold Southern Ocean. They can fly straight through waves because they have these specially adapted nostrils that allow them to not get all clogged up and drown. But we see that the hybrid offspring don't necessarily have that adaptation. We also see that the feather morphology of some of those offspring, those hybrid offspring, is probably not the best. We see like multiple feathers coming from one feather stem, and we think that probably impacts their ability to be waterproof out in the water. So we suspect that hybrids are probably not surviving to adulthood, but we set out on this project to work out what was going on there. Now, given that this is a bit more of a general session, I thought I'd drop a little bit around what conservation genomics actually is, just to keep the audience on board. We have three main goals in conservation genomics. So first of all, in the short term, we want to minimize inbreeding to avoid the negative impacts of negative fitness consequences that can occur. In the longer term, we want to minimize the loss of genetic diversity for these species. And ultimately, we're doing this so that we can ensure that the species maintains the ability to adapt to future change, which, as we all know, is becoming an increasingly important factor. So, of course, conservation genomics is just one tool in our awesome conservation toolbox. It's never treated as a single item. It's never treated as a standalone. We're always taking it into consideration alongside all of the other variables and tools that we have in that box. So our research questions for whenua diving petrels are pretty standard. We've never done any genomics for them before, and seabirds are generally very underrepresented in terms of genomic resources out there. So first of all, we were interested to characterize the genetic diversity for this species. We really want to know how genetically different they are from the common diving petrels. And we know from phylogenetics that they're not actually each other's closest species. They're not sister species. Whenua diving petrels, kuaka, are more closely related to South Georgian diving petrels. So we were interested to see what was going on there. And of course, we wanted to see if there was any genomic impact in terms of introgression resulting from this hybridization between these two species. So we set out to generate a reference genome for kuaka, whenua diving petrels, and do some population genomics work. So first off, on the genome assembly side of things, we collected blood samples in summer of 2021, I think. And we did PacBio hi-fi sequencing to generate a really good genome for that. I also did a dovetail OmniC library prep so that we could get some Hi-C data for scaffolding and build that together to a nice reference genome for our use. So just to present a little bit on the results of that genome assembly work, we had a pretty good metrics across the board for a first attempt. We've got a little cheat key there for those of you who may not be so familiar with the assembly process. The numbers we're looking at are pretty good. I do just want to make a mention, I saw a really awesome talk from Maria Akyopan yesterday that talked about reference genome bias and some of the complications of that, but I also want to say that we have a paper on using different reference genomes in birds across different taxonomic levels, and we're pretty confident that using this reference genome for these two species that are within the same genus is a pretty good way to go. So then moving on to the population genomics side of things, we sequenced 67 individuals on a NovaSeq using short-read sequencing to a target depth of around 10x, a little bit variable there. And we're aligning those data against the reference genome that we created. So we had around 38 kuaka, 38 whenua hou diving petrels, and 27 common diving petrels for this comparison. And we also had 2 individuals that were listed as unknown, the UNK there. So one of those was a putative hybrid based on its parentage, and one of those was a putative hybrid based on its morphology. So we really wanted to include that to see what was going on there. We used a pretty strict filtering for the SNPs that we extracted from that. And we were able to estimate genetic differentiation between the two species at about 0.12, which is, I mean, it makes some of those fish talks that I've seen so far look off the charts to me. But this is a pretty moderate level of differentiation. And we found that within species genetic diversity was pretty similar between these two, and I'll come back to that a little bit later. We then used DAPC, discriminant analysis of principal coordinates, to assess the differences between these individuals, see what kind of population structuring might be going on there. And we were able to see that common diving petrels cluster distinctly from whenua hou diving petrels, and our 2 unknown individuals have clustered separately from those as well. I suspect there's perhaps a little bit of over-clustering going on there, which means that they're being pulled down rather than sitting right in the middle of those 2 bubbles. But that was a really satisfying conclusion. We can see that they're clearly hybrid in terms of the genetics. So what does all this mean for the conservation of the species? So coming back to the genetic diversity measures, we found that whenua hou diving petrels, despite having this very, very small population and having really experienced an extreme bottleneck, they still have retained similar genetic diversity to the common diving petrels. But I think this is largely to do with our sampling strategy for the common diving petrels. So they occur much more widely across the Southern Ocean, but we only sampled them in the breeding location of the kuaka, the whenua hou diving petrels. So I really think that if we were to do a broader population genomic study of common diving petrels, we might come across some really strong genetic structuring, and possibly there's a lot of natal phylopatry going on there, which is driving that result. So we've only sampled a tiny fraction of the range, and so we're only seeing a tiny fraction of the genetic diversity that's present in the species. It was really nice to be able to confirm that putative hybrids really are hybrids, and that has really informed how we're managing the species down on whenua hou. It's meant that the management team are looking out for hybrid pairs that are forming, and they're trying to do some work to encourage common diving petrels to go and breed elsewhere, which is, a tough challenge. But yeah, this is really great. We've got our first baseline genomic resources for kuaka whenua hou. It's some of the very few seabird genomic resources available. I think of around 1,100 reference genomes for birds, only about 9 within this order have reference genomes available, and only 3 within the family. So this is a really big boost. Every little bit of data really helps in this space. And I'd really like to thank our iwi partners, our tribal partners down on whenua hou that provided us access to the island and have been really supportive of the work we've been doing, and all of the volunteers, sample collectors and advisors along the way. And just before I finish, I want to make a quick plug. If you ever want to come down under and you do conservation work, we're hosting the International Congress for Conservation Biology in Brisbane next year. It's going to be a great event. Proposals for symposia are now open, so please take a look. Awesome, thank you. Yes, exactly. I really want to check that we're seeing first generation hybrids, and so, yeah, structure analysis is next on my list. Cool. Any other questions? Yep. Yes, yeah, there's a whole bunch of GPS work going on right now to see where they're going, where they're feeding and foraging, and what kind of contact they're having with other seabirds out on the islands. It's a work in progress from another master's student, so yes, yeah, they're down there. They're amazing. They're so small but so fierce. Cool, thank you. Oh, so short.
Thank you, everyone, for coming. I'm going to show you some results that we have produced in our lab. I'm Henrique. I'm from Brazil, from Salvador City at UFBA, Federal University of Bahia. And the results I'm going to show you here is two systems that we have in Eastern South America, specifically in northeastern Brazil. We have a kind of island-like system within the continent. And I'm going to show you some results that we have for birds and lizards on that system. So how do species vary across the space? Well, the most common model, or the simplest model, are isolation by distance, where we have this kind of a relationship between genetic drift and the higher migration among neighborhood population. And it produces a kind of linear relationship between genetic and geographic distance. It's a classical isolation by distance model. Everyone's know that. And it produces a kind of a continuous population across the space. So it's similar kind of a stepping stone relationship. But, however, landscape are heterogeneous. We cannot see the simplest model during all times. For instance, you can see this map from South America. We do have a kind of a rugged landscape, and we can see the Andes, but we can see a lowland in Amazonia. And Eastern South America, you can see a lot of mountains. So this produces a kind of complex landscape that make barriers for species dispersed randomly across space. We also have influence from the past variation across environment. So that's the current map, but in the past we changed elevation or forest cover or expansion of dry regions. So that's all together create another models alternatively to the simplest model. So it's already proposed in literature. We have isolation by barriers, where barriers can play like rivers or a mountain can isolate populations, or resistance landscape. You can see this kind of elevation can create a resistant matrix to avoid species dispersed randomly across the space. Also environment, like soil or climate, can create a kind of barriers for species colonize all places. Altogether, we have these alternative models that we can test. So in our case of system in this Eastern South America, we try to look for two systems that we have in northeastern Brazil. The first one is on your left. It's a kind of a forest enclave that we have associated with Brazilian Atlantic forest. But we also have some influence from Amazonia in the eastern enclaves. But it's a kind of island forest in the middle of a dry region in Caatinga that we have in Brazil. If you see this Ike area, it's a kind of a Brazilian savanna. It's a Caatinga. And we have those forests just in the top of mountain. It's related to climate and some things related, and become a kind of a relictual distribution when those forests were larger in the past. We also had another system. This is a Restinga system. It's also associated with the Brazilian Atlantic forest, and we have this kind of a shrub vegetation just close to the sea and associated with sand dunes. In both systems, in northeastern Brazil, we have a kind of island. I'm going to show you in details each of them and the results that we have. So for the first one, in the forest enclaves, we try to see if the climate change from Quaternary drove some differentiation or connection among enclaves, and how the features of landscape drove the population differentiation across space. We choose two species for that. Both are endemics from the enclaves. are the Conopophaga ceari and Sclerurus cearensis. They do not occupy all the enclaves. That there are many reasons for that. I'm not going into details for that. But they have distribution in some enclaves. So we try to see if this isolated population enclaves can show some population genetic differentiation. We use subegenomic sampling using DD-RAD for both species. And for both, we found population differentiation. If you saw PCA plot, we can see that most of enclaves are pretty much differentiated. And when you try to see the population differentiation using SNMF, we found just 3 clusters for each of them. For the Conopophaga, we found a southern cluster and an eastern and western cluster. And for Sclerurus, we've also found a kind of a little bit eastern cluster and a middle and western cluster. We also found some enclaves that we have a kind of admixture between some clusters. You can see, for instance, in Baturité for Conopophaga ceari and Uruburetama for Sclerurus, we have a kind of admixture. It seems that we have a kind of a mixture of colonization during the expansion of these forest enclaves. I'm going to show you in more detail in the next slide. So we also did some paleodistribution across the late Quaternary. And you can see during the period calling Bolling Olerod, we have a higher distribution of the enclaves when compared to the present distribution. It seems that those enclaves were higher in the past than now. It's also associated with this climate change here. Just in the Bolling Olerod, we have an increase of humidity in those regions. So it's a lower kind of a forest expansion and might have a lower species to colonize or enclaves or also expand the population size. To confirm that, we also, so it's not yet, but we also calculate some migration surface to see if those barriers across the spaces. And we can see for Conopophaga, we do have barriers associated with valleys across the enclaves. You can see in the arrows here, we have a valley just enclosed, population are close in space, but they have a huge genetic differentiation. So there is no elevation to connect those enclaves. It also happens for Sclerurus. We do have here. And we also have distant population that has higher levels of migration. And you can see that those populations are connected kind of mountain channel, which allow the population or forest enclaves expand across this elevated region and provide population connection. We compare different features of landscape and how those features can influence population differentiation. We use GDM, generalized dissimilarity modeling, and we use some predictors for IBR, the kind of elevation. We also use the classical isolation by distance and isolation by environment. We use bioclim variation. And we also use a kind of a paleo-IBR. We got those distributions, the higher distribution in our paleo modeling, and combine all of them to see which one can predict the genetic differentiation across enclaves. And you can see for Conopophaga, the significant distribution was isolation by distance. We didn't see other significant results. And for Sclerurus, we have the paleo-IBR, this resistance matrix from our paleo distribution. But one point interesting, those predictors just explain a little bit of genetic variation. It seems that there are a lot of genetic variations not explained by the predictors. So there's much more happening that we can see with our genetic landscape modeling. We also did some historical demography just to confirm with this higher distribution that we saw in niche modeling also happened for in genetic data, and you saw that. You saw kind of synchronic decline in all population starting about 10 or 15,000 years ago for both the stairway plot and also some simulations using fasting synco. You can see that the most probability, the most probable modeling model was the decline of all populations. So in that case, we saw a kind of a combination of historical distribution plus current features of landscape driving the differentiation and also connection among those isolated enclaves. In the other model that we studied is the Restinga for the lizards. We do have this system. It's a kind of small regions, about 400 kilometers from Salvador until the mouth of San Francisco River. I don't know if everyone knows that. But across that region, we have this kind of islands of sand dune vegetation. We do have this small sands, white sand dunes. It's from Pleistocene, a kind of old sand. And we have this shrub vegetation where we have some endemic species of lizard. We have Tropidurus zigomi. It's a species distribution across the whole enclaves. And the other groups are kind of species complex of Glaucomasticus abaetensis and itabaianensis. They are sister, and was just one of them was described recently, but it's a kind of a complex. And we use these two species to compare the population variation across this kind of island-like system. You can see in the photos here, the system are the sand vegetation are not continually across the space. You have a kind of islands isolated by mangroves. You can see in that photo here. So for that group, interestingly, we found deep genetic variation across the whole distribution. We found 8 clusters in each group of lizards, and they're pretty much concordant, we can see. And almost each population that we sample is a kind of a cluster, really differentiated. We can see the SNMF and SVD quartets. All populations are differentiated, but we also have some admixture in some population. We can see this population from Praia do Forte. They have the same admixture for both groups, and for one species we also have admixture in Baixio region. But one point is this population differentiation is pretty much concordant with this island of sand dune vegetation that are isolated by river mouth. We also did this migration surface to see the barriers, and you can see some barriers are more stronger than others, especially that one in the Itabaiana hills is a kind of a relictual population, or sorry, a relictual Restinga that we have inside the continent. It's the red one cluster for both species, and it's a pretty much differentiated cluster in both groups that we saw. It's interesting because a small population is pretty much differentiated and is not connecting with all population across the coast. And for that group, we also saw population decline for all clusters. I'm just showing a couple of them here, but in all of them, we have a pretty much similar pattern between 20 and 10,000 years ago. We are still doing some simulations with fasting core. It's not done yet. But for that group, we also saw kind of a combination of current landscape features and historical features of landscape. Because in the past, the Restinga region has a chance to occupy a higher portion that they have now. We can see in this ecological niche modeling for 17,000 years ago, we have exposure of continental platiform, and maybe the Restinga load might have advanced across this region and also increase the population size for that group. So this combination of historical and current landscape features also drove this lizard groups. And we have, for different systems, have the both scenarios. Interplay between historical and current features of landscape influence the population differentiation. So I would like to thank my collaborators and students that also did some of the results that I'm presenting here. And that's it. Thank you. And if you have questions. Oh, sorry.
Great talk. Could you talk a little more about how do you lead your isolations, IgG, IPR, IPE?
Yeah,
a little about
I just talked. very fast about that, but I compare those models using generalized similarity modeling. With that model, you can use a matrix of population differentiation, and you can set up all the predictors that you have. You can combine matrix or linear variable, and then this model fits the predictors to the response variable using a nonlinear relationship. And you can also do a kind of variance partitioning to see how those predictors can, or how much of that predictors can predict our genetic variation across the space. Yeah. Okay, no more times these questions. Thank you.
Hello, I'm Brian Dillard. I'm part of the Muller Lab at Cornell University. And today I'm going to talk to you about a bit about the work we've done, kind of looking at the changes that we see when we take wild mice and look at their gut microbiota kind of 100 years in the future, I guess now, and we compare that to like laboratory mouse gut microbiota. So first, like the first question is, why are we looking at gut microbiota in general? And the easy answer to that is that most mammals have a gut microbiota and that they're deeply integrated. This is just kind of a little cartoon here showing some of the effects or some of the ways in which the gut microbiota interacts with their host, whether or not that's like producing short-chain fatty acids and modulating gut homeostasis or kind of conditioning the immune system. And when we see breakdowns in these really important interactions, we see negative outcomes, right? So whether or not that's degrading host physiology or changing host behavior. And one place we see these negative effects is when we look at the differences between lab and wild mice. Previous work has shown that even though lab and wild mice harbor kind of distinct sets of bacterial strains belonging to a shared set of taxa, differences cause changes in immune cell proliferation, infection resistance, cancer progression, and one of the main reasons that we've chosen to look at this is the ability to model drug outcomes in humans, right? We use lab mice in so many tests, so many clinical trials, and when the results of lab mice don't kind of mirror those found in humans, we start to, those results are no longer valid necessarily. So today, I'm going to first talk to you about the discovery of ancient symbioses in inbred lab mice. Then we're going to talk about the genome-wide signatures of selection and some interesting results with genetic drift. And then we're going to look at kind of the fitness of lab strains compared to wild strains, and whether or not these lab-specific adaptations can maybe outweigh some of the negatives brought on by increased genetic drift. So one of the first things we have to think about when we're looking at lab gut microbiota is kind of the origin, right? So over 100 years, lab microbiota has diverged in composition and taxonomy. And the question is, are these laboratory-associated microbiota derived from humans, the laboratory environments, their new, nice diet, or have they inherited some of their microbiota from wild ancestors? One of the ways we can kind of look at this is through co-phylogenetic analysis. Here is an example from the primate gut microbiota. On the left side is a host tree from some primates, and on the right side is their associated gut microbiota. We can see that in this case, the Prevotella species, the human Prevotella species is directly across from the Homo sapien, or the human host tree. So we see that in each of these cases, the gut bacteria derived from the host kind of mirrors their phylogeny. So they've co-diversified over time. In this case, it's over the course of 25 million years. So for this study, we've assembled kind of the same data set. We've assembled 5,567 metagenome assembled genomes, or MAGs, from 300 individual rodents, represented by 14 different rodent host species. Using short and long read sequencing, we can then create a huge phylogeny where each tip is a bacterial strain, and then the outside ring kind of indicates the phyla of each strain. If we kind of scan this tree or scan the distal 1/4 of this tree for evidence of codiversification, so evidence of codiversification of the bacterial lineage with their rodent host, we see that about 25% of this tree shows this evidence of codiversification. And then if we kind of subset this data to look at only the codiversifying clades that are ancestral to murids, which includes our lab mice and our wild mice, we see that there are 24 phylogenetically independent codiversifying clades retained in laboratory mice. So I guess what this means is that lab mice have actually retained more than half of their ancestral codiversifying gut bacteria, right? So even though lab mice have been in the lab for about 100 years, there's still some lineages of bacteria in their gut that are very, very closely associated with wild mice and have kind of followed them over evolutionary time periods, right? So we see that. Now the question is, like, in what ways have these bacteria diverged, right? Like, these bacteria are very closely associated with their host. And if we look at some of the actual cophylogenetic analyses, so in this case, the bacterial lineages are on the left and the host tree is on the right. We can see some of these associations. And focusing more on the left tree, the red tree, if we look at these arrows, we see that these wild lineages cluster together and that the lab lineages cluster together as well. And if we look at the branch lengths, we see that there is some amount of diversification, right? So these lineages have kind of evolved over these 100 years. And one of the questions is, like, what evolutionary forces have driven this divergence over time? One kind of obvious answer is natural selection, right? These strains are in a lab mouse, and these lab mouse are exposed to lab diets. They have very inbred genetic backgrounds. They're very nicely cared for, and they have very different just whole lives, right? On the other hand, though, the deriving a lab mouse strain and keeping them in the lab for 100 years comes with some pretty significant population dynamics, right? We see founder effects, we can see bottlenecks, and all of these things can kind of impact genetic drift over time. So to kind of tease apart these two kind of modes of evolution or types of evolution, we can use DNDS, which is a ratio which quantifies the rate of protein evolution compared to the ratio of evolution at silent sites. A DNDS of less than 1 is a sign of selection to keep the protein the same, or purifying selection, and a DNDS of greater than 1 is a sign of positive selection, or that, like, the protein sequence is generally changing, right? In many, many cases, in almost all genomes, you expect most of the genome to be under pretty strict purifying selection, right? Like, most genes are doing what they need to do, and any changes to those genes can usually lead to decreased fitness or deleterious or death, right? So looking for natural selection, we expect, like, this is kind of where we expect to see, like, adaptive evolution, right? So we expect certain genes to be under high degrees or high levels of DNDS, right? So DNDS of over 1. So this is pairwise comparisons of lab genes and wild genes and their DNDS. The y-axis, so the left side, is the DNDS in lab, and then the x-axis is the DNDS of wild, right? And like I said, most genes are under purifying selection, so that's kind of what this bottom left cloud denotes. So this is a DNDS of less than 1 in lab and a DNDS of less than 1 in wild. This point cloud in kind of the top left portion is what we're kind of mostly interested in. These are the genes that are under positive selection in the lab, but purifying selection in the wild. And these are all the targets of potentially that adaptive selection, right? The two guys on the bottom, PGI and PVFLB, are both having to do with carbohydrate metabolism. CARB-B2 is a antibiotic resistance protein or gene having to do with, it's a beta-lactamase, so confers resistance to something like penicillin. And the rest are kind of have to do with various cellular processes like tRNA replication and stuff like that. And there are genes under positive selection in wild, but in general, genes displaying a DNDS above 1 are enriched in lab relative to wild, right? So we see a kind of a significant increase in this adaptive evolution in our lab mice. So we see evidence of natural selection, which is not super surprising. But what about genetic drift, right? Here we can again use DNDS, but instead of using DNDS of individual genes, we can kind of look at an average or genome-wide DNDS. And this DNDS reflects kind of demographic processes, and it has, at least in bacteria, a long history of being used as a proxy for something like effective population size, right? And if we see higher genome-wide DNDS, this is generally indicative of, like, increased mutational burden or increased genetic load, both of which kind of would lead to, like, a reduced level of fitness. These are density plots. You can read them similarly to a histogram. The gold is the wild and the blue is the lab. And in general, we see our lab mice are shifted up to the right, which indicates like a higher genome-wide D&DS relative to their wild counterparts. The top left plot is just all of our co-diversifying strains, and then if we actually look at strain or as essentially clade by clade, we see that there are some lineages that show like kind of a bigger effect size, right? So here we see that genome-wide D&DS is increased in lab relative to wild. Okay. Oh, and that elevated genome-wide D&DS, so the same is not found in non-codiversifying clades, right? So we look at lineages that are not codiversifying, are found in lab and wild, this increase in D&DS is not apparent, right? So this is only in these like ancient symbionts. Okay, so we have these ancient symbionts. We've seen that these ancient symbionts are adapting to their host, but they also have kind of these higher levels of genetic drift, right? And the question is whether or not these lab-specific adaptations kind of outweigh this genetic drift. And we can look at this through like a competition experiment to get we used data from a previous experiment that co-housed 3 types of mice for 17 days, those with the wild microbiota, those with the lab microbiota, and then germ-free mice. And the whole idea is, are the lab or wild strains better at colonizing these germ-free mice? So just a competition experiment. And in general, we find that these wild bacterial strains that aren't under this increased genetic load outcompete phylogenetically matched laboratory bacterial strains in every single case. The y-axis of each of these plots is essentially abundance within the germ-free mouse, and then the y-axis is the number of days, right? So in every single circumstance, we see that these gold wild-derived mice or wild-derived lineages are better able to colonize our germ-free mice. That's pretty much it. So in summary, laboratory mice still harbor 24 codiversifying clades and ancestral lemurids, and that's generally half of the codiversifying clades found in wild mice. There are some genes under high levels of positive selection, but the kind of more interesting result is the genome-wide elevations of drift, or at least in my opinion. And then these wild-derived codiversifying microbiota outcompete their laboratory counterparts, showing that this drift or this genetic load is reducing fitness enough to have this like kind of fitness differential. Yeah, that's it. Just want to acknowledge my lab and my co-authors on this paper, and I'll take any questions. Yep.
In the genes that you found positive selection, were these in essential genes or just like other genes or in like that you found within each microbiome?
Yeah, so it's hard to tell whether or not they're essential. I mean, there are genes that were involved in carbohydrate metabolism, which I wouldn't say is super duper essential. Like there's lots of sources of energy. It's hard to tell if they're essential or not. Yeah, sorry. Any other questions? Yeah.
So the wild ones colonize better, or do they perform better on the lab diet?
Ooh, that's a good question. So I guess in this case, they're on the lab diet as well, because they're essentially co-housed. I guess maybe I'm not understanding the question.
Well, I guess you could like give some only wild, give some only lab, and then
Like how that like. So that is essentially done. So well, maybe not quite like that, but yeah, no, that would be interesting. So if you take a germ-free mouse and give it a wild microbiota, that mouse is more fit than if you give it like another rodent microbiota. So I'm sure something similar would probably happen if you did that same, but with wild and lab. In fact, it might have been done. Maybe I'm just blanking on the literature. Yeah. In the back, maybe first. Yeah. Yeah.
Among the same genes that undergoing positive selection, you find any genes related to social behavior of the mice?
So, not directly. It's hard to know like specific genes that would change host behavior, specific bacterial genes at least that would differ or change host behavior. But that is definitely something interesting. You can definitely, Madeleine over here in our lab does a lot of host behavior analyses with regards to the gut microbiota. So I'm sure you could do some complex analyses. yeah, we haven't necessarily, because yeah, we have that whole figure of all the ways in which microbiota impact the host, but we haven't necessarily looked at the specific bacteria and the specific genes and how it relates to that yet. That's a good idea. Cool. And did you have a question? Perfect.